Hey everybody, welcome. So we're going to be covering Unit 2, Cell Structure and Function, specifically Section 2.2, which is Cell Structure and Function. If you're confused because the unit is the same as the section, that's okay. It is a tad bit confusing. What you need to know is this is an extension of 2.1, so it gives a little bit more details about very specific subcellular components. Please note, if you haven't reviewed that video, it might be in your best interest to go back and review Unit 2, Section 2.1, which is about subcellular components. If you haven't done so, we have the video listed in our YouTube page. And if you have, congratulations. We're going to continue on with a little bit more details with that subsection. Please note the disclaimer below before we begin, and let's get started. Okay, let's get started. But before we get started, I want to provide you a brief history lesson. And this history lesson is going to take you back to a period of time before even humans existed, even before dinosaurs existed. So I'm not really good with history, so I can't give you a specific time frame. This is why I am someone that's in STEM. But for the purposes of this video, you just need to understand this is a very, 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 very long time ago. And during that very, very, very long time ago, this is before even oxygen existed. There was no O2 anywhere. And during that time, it was believed that there were these cells and they were just free floating everywhere on planet Earth. And they didn't need oxygen to survive, and they didn't have membrane-bound organelles. So they didn't have all the organelles that we talked about before. So they didn't have the Golgi apparatus. They didn't have the nucleus. They were just free-floating cells. And you could think of them being simplistically prokaryotes that were just very simple, just free-floating, and they just existed. And what ended up happening, for whatever reason, evolution, at some point, we did have oxygen. And oxygen existed, and these cells needed to find a way to deal with the oxygen to be able to survive in that environment. And some of these prokaryotes, it was believed, ended up dying off, but then some of them ended up surviving because they evolved to have organelles or be a specific organelle that was able to sustain oxygen. So basically, they were a type of cell that was able to sustain the environment of oxygen. And we believe that now those are our mitochondria, that they weren't in a cell that we know as now, they were just free floating by itself, and they were able to sustain itself with understanding and living in an environment with oxygen. So that's why we believe, or we do know, that their mitochondria currently has its own DNA that comes from your mother. It's a maternal based organelle. So anytime you take a deep breath, you're breathing essentially because your mitochondria work, and your mitochondria work because your mother provided you with her DNA to allow you to have mitochondria. So that's just a little fun fact for you. But that mitochondria now was able to sustain itself in an environment with oxygen. Because it was able to sustain itself in an environment with oxygen, we still had these prokaryotes that were floating around and mitochondria. And it was believed that at one point there was a mitochondria that ended up engulfing a mitochondria. So there was a prokaryote, I should say, that ended up engulfing a mitochondria. And that is how we have eukaryotic cells today that have mitochondria. That is the theory. It is called the endosymbiotic theory. And that is how important mitochondria is, that we believe that at some point prokaryotes have evolved to eukaryotes because there was the creation or development of mitochondria, and a prokaryote ended up engulfing a mitochondria, which then became a eukaryote. So that's how important mitochondria is in the terms of biology. And that theory comes up every once in a while. You don't need to know the nitty-gritty, you just need to know the overarching theme to help you remember that mitochondria is a very important organelle, especially for animal cells. So just a note, when we talk about mitochondria, we're talking about it being in animal cells and comparing it to chloroplasts, which are found in plant cells. I also want to note that while mitochondria are in animal cells, they have been found in plant cells as well. So they are not the primary function of plant cells, but I just want to know that this is something important to note, and we're going to talk about it in just a second. So when we talk about the presence of where mitochondria is versus chloroplasts, mitochondria is found in all eukaryotic cells. And remember, when we divvy up or we divide eukaryotic cells, that is both plant and animal cells. And this is how they trick you in questions, because your tendency is to think that, okay, if I'm talking about an animal cell, of course, that's a eukaryotic cell, but a plant cell must not be a eukaryotic cell. That's what you're thinking, but that is incorrect. You now know that eukaryotic cells are both animal and plant cells, and mitochondria exists in both plant and animal cells. So if you ever get a true-false question, you'll know that for certain. And chloroplasts are found only in plant and algae cells. So that's something to know about the presence. So let's go to functionality. For mitochondria, they are primarily used in animal cells, and we utilize them to breathe, cellular respiration, because we take our food, we don't sit out in the sun and consume our energy. Instead, what we do is we break down our energy through food, which produces ATP through mitochondria. So mitochondria is considered the powerhouse of the cell. In opposite, chloroplast utilizes the light. So plants stay outside or they stay inside and have a light source, and they go ahead and convert that light energy into some sort of chemical energy like glucose. So you could think of something like spinach or something like a vegetable that you consume. It has sugars in it because it's able to convert that light energy into chemical energy. In terms of structure, we're going to talk about this more in detail in just a second, but they both have a double membrane and they both have individual structures that are specific to the specific organelle. In terms of pigmentation, chloroplasts, the green pigments, that's chlorophyll. In terms of DNA for mitochondria, contains its own DNA. Chloroplasts do too, so that's a similarity. And then ribosomes, they both have ribosomes. The process, we talked about this before, for mitochondria, it's cellular respiration. Chloroplast, it is photosynthesis. Energy source for mitochondria, we consume simple sugars, or we consume food, which has sugars in them, like glucose or fructose, whatever. And we use that as our energy source when it's eventually broken down into glucose. And then for chloroplasts, it consumes sunlight. The products of mitochondria, or what's being released, is ATP, CO2, that's why we breathe out carbon dioxide, and water, which we release. And chloroplasts have the opposite, where they produce glucose and oxygen. That's why it's good to have a lot of plants, because they release oxygen that we as human beings can consume. Let's talk about the mitochondria and the picture on the right here. Mitochondria, just like chloroplasts, have two main membranes, the outer membrane and the inner membrane. Together, they work together to formulate the shape of mitochondria. Now, there are two additional parts with that. With the inner membrane and the outer membrane working together, there are these folds that exist. So this here, or you could see some here. Those folds are called cristae. And cristae work to formulate or create more surface area. I want you to think of a 3D shape and the fact that we might have to take the 3D shape and cover up the outside. Maybe we want to gift wrapped or something like that. So we want more surface area to help wrap. And that's what surface area is. The purpose of the cristae is to increase that surface area to provide more space so that the process known as the electron transport chain, or ETC, can take place. And the reason why ETC is important, which you'll cover in a later unit, is so that it can produce ATP. And ATP is important because it is crucial for survival. It is our energy, and that's why we consume food, to produce our energy. So it's the important part of cellular respiration. It's one of the steps. So that's why it's so important. The other part is the matrix, the other mitochondria part. And the matrix is noted to be the site of the Krebs cycle, which is another part of cellular respiration. So it's the precursor of electron transfer chain, and it helps assist in making sure that electron transfer chain takes place. So because you have the matrix, you have the Krebs that takes place, which also means that if it works more efficiently, then the electron transfer chain can exist. And because it can exist and work efficiently, it produces more ATP. And if we have ATP, we have more energy, which is important. So the main purpose overall is that the parts of the mitochondria work together to produce ATP, which is a main part of cellular respiration, which we'll cover in another unit. Now, with chloroplasts, you want to think of the primary color being green. It has that green pigment, and that is because it has what we call chlorophyll. And so chlorophyll is important for chloroplasts. It's what gives it that green pigment in plants. So they work together to basically harness the sun's energy, which is light, for the process of photosynthesis. So unlike cellular respiration for mitochondria, chloroplasts are mainly for photosynthesis. Now let's talk about the shape of chloroplasts. So they have this unique shape. They have their own ribosomes as well, just like mitochondria does. And they have inner structures, which are important. And they too work together to increase surface area, so that way it could harness the sun's energy to then therefore produce food. So the opposite. So you're basically going from ATP to then going to create sugars, which we'll talk about again in a later unit. So with the shape of the chloroplast, it is important to note that you have what's called thylakoids. So this is the thylakoid. The thylakoid has a disc-like shape. And if you zoom in, that disc-like shape is flat. And the reason why it's this flat shape is so it has more space to capture that sunlight and convert. When you have a stack of these, so you have a stack like this, that's called a grania or granium. One is singular and one is plural, but basically it's just a stack of thylakoids. So that is where we go ahead and take that sun's energy, or ATP, and harness it. Okay, so now we talked about the thylakoid, we talked about the granium. Now around, there's all this fluid. And that fluid holds the chloroplast basically in its structure. And it's also the site of what's called the Calvin cycle, which is a part of photosynthesis. It's one of the steps of photosynthesis. So the stroma is the fluid around. Now, we talked about all the different parts. We've talked about the granium, the stroma, the thylakoid, and internally, if we were to take like a cut of the disc, we would call that the thylakoid membrane, the inside innard part, that yellow part right there. And just as a reminder, we stated this before, but just like mitochondria, there are a double membrane. There is a double membrane. So there is an internal membrane and an external membrane and an inner membrane space that is there for chloroplasts. So let's talk about lysosomes and peroxisomes and go back to section 2.1, where we have a cell and we talked about the Golgi apparatus. The Golgi apparatus acts as the post office where items enter and exit the cell through the Golgi apparatus. It's the post office of the cell. And the way they enter are through these circle-like structures called vesicles, which provide a barrier, like a balloon, against the internal substance, whatever is inside, against the outside substance, the cytosol or cytoplasm. So there are two types of vesicles that we tend to reference, and those are lysosomes and peroxisomes. When I think of lysosomes, I think of the root lyso or Lysol wipes, and it helps me remember the functionality that it's used to digest enzymes and process waste. So it is there to process the waste before entering that Golgi apparatus. Then peroxisomes, if you think of oxy, you think oxygen, and detoxify also has oxy in it, or OX, and I should say OXI and OXI, like oxygen, and oxygen will help you remember that peroxisomes are mainly for detoxification and breaking down of fatty acids. Now let's try to figure out a quiz question. This is a critical thinking question. I want you to take a moment, read this question. Once you're completed, make sure that you come back to this video. At this time, it might be best to pause this video, take about a minute or so to read the question, and unpause the video once completed. Please note that this question should take no more than 30 to 60 seconds. So go ahead and pause your video now. Okay, now that you have read the question and reviewed your answers, we're going to see if we could try to figure out the answer together. Did you select all of the above? If that was the case, you would have had the correct answer. So let's take a look at the question together. It says, given the endosymbiotic theory, which proposes that mitochondria and chloroplasts were once free living organisms, which of the following best explains how these organelles became so integrated within eukaryotic cells? So this is the type of question where they talk about eukaryotic cells, and they want to trick you because they want you to think that we're differentiating between eukaryote versus prokaryotic cells, and that is not the case. What we're taking a look at is mitochondria and chloroplasts and the similarities and differences in terms of understanding animal cells and plant cells and their main functionality. Now, in the first part, it says the organelles developed a symbiotic relationship within the host cells, exchanging essential nutrients and energy. The reason why this is true is because mitochondria, remember, can produce ATP. And the thing is, the mitochondria producing ATP can be then utilized for chloroplasts, which takes the ATP and produces the sugars that we require. So this is a very convoluted statement, and the understanding is that there is a symbiotic relationship, and you might be like, this is not a term that I understand. We didn't talk about it, and we're just in unit 2. Whoa, whoa, whoa. And that's okay. You need to deduce, based on the information that you already have, the fact that a symbiotic relationship is exchanging information. There's a back and forth between mitochondria and chloroplasts, which is what they're trying to say. And the reason why this is true is because you have to think about it in terms of animals versus plants. And the bigger theme here that we talked about in this subunit, so section 2.2, is that we said that mitochondria goes and is in animal cells. We consume our food. We consume plants as our sugar, maybe directly or indirectly, but we consume some sort of plant, and that utilizes chloroplasts. So we depend on chloroplasts because they provide us the nutrients that we need in mitochondria.
we go ahead and produce the sun or the ATP that the sun harnesses that chloroplasts need to then produce the sugar. So it's a give and take relationship. And so that's why A is correct. If I look at answer choice B, it says the host cell engulfs the organelles, but they maintain their independent functions. So that is what we talked about the endosymbiotic theory, the overarching theme, the understanding that at some point there was a mitochondria that had evolved from a prokaryote and that the prokaryote had engulfed that mitochondria and that ended up becoming a eukaryotic cell. However, remember, we said that mitochondria has its own DNA maintained from mother. And so that's what this statement is saying. You know what? The mitochondria, yes, was engulfed, and that's what the endosymbiotic theory is, but it's very independent. It's very much on its own because it has its own DNA, which if you look at the previous table, the first or second table that compares mitochondria to chloroplasts, you'll notice DNA independence is very important. It is an organelle, both of them are organelles, are very independent because it has its own DNA. And then statement C says over time, the organelle transferred most of its genetic material to the host nucleus. While we don't have this information privy to us, it's not something that we covered so far, which you'll find in AP biology related questions. You will note that part A and part B were correct. Overall, we do know that there is genetic information that has been passed along and that transfers over. You don't need to know that information right now for this specific question. This is a critical thinking question, and that you should have known that A and B incorrect. Therefore, more than likely, D would be a correct answer. But for statement C, this would be a statement that you would understand later on down the line. So this is a critical thinking question to think outside the box, to have you understand how endosymbiotic theory is and the relationship between mitochondria and chloroplasts. As you notice, the questions are not completely direct. They are based on the information that you gathered and the knowledge to think outside the box. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams. We can't wait to help you succeed.